Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is the two component signaling pathway or signaling system. Okay, so before going to this topic, I want to say that please you share and subscribe this channel for the growth of this channel, and if you will try to provide your support for the increasement of the growth of this channel, then I will give you my best for you. Okay. So here the signaling system, that is the two component signaling system. It is the most common form of the signaling pathway that responds to extracellular events in bacteria and plants. So where this type of component signaling system is seen generally in the bacteria and the plants. Here the two components, these are involved in the signaling system. Okay, and it consists of a sensor. One is sensor and another is the regulator. Two important actually part, you can say two important part that are involved here for continuing the signaling system, that one is sensor, another is the regulator. So what is the function of the sensor? Sensor actually is an autophosphorylating histidine kinase. Okay, this is the autophosphorylating histidine kinase. This is the sensor. And the regulator is that this is the response regulator. And what is the function of this response regulator? That transfers the phosphate group from the sensor kinase to the conserved aspartate within itself. That means it helps in the transformation of the phosphate group. So what's the function of the sensor and the regulator? Sensor that involved in the autophosphorylation process, and regulator that transfer the phosphate group from sensor to the aspartate that present within this. And the sensor histidine kinase, generally it is located where? It is located in the membrane, and it can be activated when it can be activated by binding of ligand, and that is in the extracellular medium. So when that remain on the membrane, what is remain on the membrane? That is the sensor histidine kinase, and when ligand bind with this sensor histidine kinase, then it becomes activated. Okay, so activation that causes the kinase to autophosphorylate. So that means when the ligand that binds with the sensor, then the autophosphorylation process starts, and the reaction transfer the phosphate from the ATP onto a histidine residue in the kinase domain. So here the autophosphorylation process, after the autophosphorylation process, the transfer, the reaction, this autophosphorylation process helps to transfer the phosphate group from where? Phosphate group from ATP to the histidine residue. Okay, so here the transfer of phosphate group from ATP to the histidine residue is occurring, and that induced by the autophosphorylation process. Okay, that is induced. And the sensor here interacts with an effector protein, and one effector protein also involved here that interacted with the sensor, that is interacted with the sensor, and this effector protein is the response regulator. Okay, this response regulator has two domains. One is the conserved receiver domain and another is the effector domain. And what are the role of these two domains? That first one, this catalyzes the transfer of the phosphate group from the histidine of the sensor to the aspartic acid, and this activates the two effector domains. Okay, so here first is the conserved receiver domain. That is the main important domain because it helps in the catalyzing process of transferring of the phosphate group from histidine to the aspartic acid. And due to this catalyzation process, the second effector domain that becomes activated. Okay, the effector domain that becomes activated. So here are two effective domains are present. So that's why we have to write 2. And this is the very rough sketch that here the sensor histidine kinase and another is the response regulator. Both are interdependent on each other. If sensor histidine kinase that becomes phosphorylated, then the transfer of phosphate group is occurring from the sensor histidine kinase to the response regulator, and after this the signaling process is continuing. Any signaling process that is continuing. Okay, so this is the two component signaling system, and it is very short and short description you can say, or short topic, that is how the sensor and the regulator both are interacted with each other, or both are interdependent with each other for continuing the signal process. Okay, so here two are involved, and that's why this is called the two component signaling system. Okay, so you just read from any cell biology book and after seeing this video. Okay, so thank you, and I will try to explain another topic in my next video. So continue with my channel.